There is a version of this story that sounds like science fiction. A person hears a sentence. Their brain activity is recorded. A model looks at the recording. Text comes out. If you stop there, the headline writes itself. Machines are learning to read minds. But that headline is exactly the wrong lesson. Brain recording matters because it shows that non-invasive recordings of brain activity can contain enough structure to support typed sentence decoding under specific experimental conditions. That is real progress. It is also not telepathy. It is not arbitrary thought decoding. It is not a clinical communication device that has been demonstrated for patients. And it is not proof that the signal itself has become an interface. The real story is more interesting because it is more demanding. A brain signal does not arrive already translated. It is measured through hardware. It is shaped by the task the participant is performing. It is filtered through the quality of the recording. It is interpreted through training data, calibration, decoding models, language constraints, feedback conditions, and evaluation metrics. It sits inside consent, privacy, usability, and deployment questions. So the question is not simply whether information can be extracted from the brain. The question is, what kind of system has to exist before that extraction becomes a practical brain-computer interface? Brain 240 gives us a useful case because it is neither trivial nor magical. In the first version, the work established a peer-reviewed foundation for non-invasive typed sentence decoding. Healthy volunteers typed briefly memorized sentences on a QWERTY keyboard while their brain activity was recorded with EEG or MEG. The final Nature Neuroscience record reports that MEG substantially outperformed EEG. Average character error rate was lower with MEG, and the best MEG participant performed better still. That separation matters. MEG and EEG are both non-invasive, but they are not interchangeable. MEG measures magnetic fields associated with neural activity and, in this study context, produce stronger decoding performance. EEG measures electrical activity from the scalp and is cheaper and more familiar, but the reported performance was not the same. A result from MEG cannot be casually turned into a claim about consumer EEG. A result from healthy volunteers typing memorized sentences cannot be turned into a claim that patients can now communicate freely through a clinical device. The second version is the current trigger. Brain 240 v2, still marked as under review in the controlling source materials, pushes the system towards sentence-level decoding from continuous MEG recordings. The study involved 9 healthy adult volunteers, all proficient typists, recorded across roughly 10 hours each. Participants listened to sentences, waited through a delay, and then typed the corresponding text. The model was not watching ordinary life, private inner speech, or spontaneous conversation. It was decoding a structured language production task. That is the first boundary to keep in view. What was measured was brain activity during a constrained typing-related task under MEG recording conditions with a known experimental protocol. What was predicted was typed sentence text evaluated against the study target. What was not demonstrated was general access to thoughts, intentions, desires, or private mental content outside the task. The architecture matters too. Brain 240 v2 is not a single magical artificial intelligence system staring at a brain scan and discovering meaning on its own. It uses a multi-component decoding pipeline, a character-level encoder, a word-level alignment mechanism, and a large language model adapted to integrate MEG-derived information with text-like intermediate signals. The language model helps resolve structure, but it does not independently read thoughts. It operates inside an engineered translation system, constrained by the task, the data, and the model design. That distinction is crucial because language models are good at making incomplete signals look coherent. If a model produces fluent text, audiences may assume the brain recording carried all of that meaning directly. The safer interpretation is narrower. The system combines neural signals, learned task structure, and language regularities to improve decoding under the tested conditions. The result is impressive because MEG-derived information contributes to the decoding. It is not proof that the model has solved general brain-language causality. The performance numbers need the same discipline. V2 reports an average word error rate around 39%, often expressed as about 61% word accuracy, with stronger performance for the best participant. Those numbers are meaningful in context. They are not a clinical communication rate. They do not say how well a patient who cannot type would perform. They do not say the system works in real time as an everyday assistive device. They do not erase the latency, hardware calibration, or patient transfer problems that the source material itself keeps visible. This is where the interface stack becomes the center of the episode. Signal acquisition comes first. What sensor is used? MEG and EEG do not give the same signal quality, the same cost profile, or the same deployment reality. A 306 sensor cryogenic MEG system is a very different object from a wearable consumer device, a hospital tool, or a home assistive interface. Signal quality comes next. If the recording is noisy, variable across people, or sensitive to movement and environment, the decoder has to work harder. A brain-computer interface cannot be built from an idealized signal. It has to survive the signal people can actually produce and the hardware can actually capture. Then comes task and context. Brain 2 QWERTY is about typed sentences under a known protocol. That is not a weakness. It is how the experiment becomes interpretable. But it also means the evidence is not about arbitrary thought. A person performing a structured typing task is not the same as a person silently wanting to communicate anything at any time. Training and calibration follow. A practical interface has to handle people, not just data points. It has to adapt to the user, learn the relevant patterns, and manage variability across sessions and individuals. The V2 limitations make this visible. Inter-individual variability and missing key presses for target patient populations are not side notes. They are central translation problems. Model decoding is only one layer. The decoder has to transform recordings into useful outputs, but usefulness depends on feedback, error correction, latency, and confidence. A system that produces a plausible sentence too slowly or without a way for the user to correct it is not yet a working communication interface. Reliability and usability are another layer. Can the system work repeatedly? Can a user tolerate it? Can errors be detected? Can the interface communicate uncertainty rather than simply outputting fluent wrong text? These questions matter more as decoding gets better, because a more convincing output can be more dangerous if the surrounding interface is weak. Privacy and consent are not decoration. The V2 study reports written informed consent, compensation, local ethics approval, and de-identification or anonymization before analysis. Those facts matter. They support a responsible experimental frame, but they do not answer every neurodata governance question. They do not decide who may reuse recordings, what future inferences are permitted, how consent travels across contexts, or how a clinical need should be protected from consumer hype. Deployment is the final test. A laboratory decoder can be scientifically important while still far from practical deployment. To become a clinical brain-computer interface, the system would have to work under the constraints of the people who need it, people who may not be healthy, proficient typists, who may not produce ordinary key press targets, and who need reliable communication rather than a demonstration metric. This is why Brain 2 QWERTY should not be dismissed. It is a strong result precisely because it makes the hard parts clearer. It shows that constrained non-invasive brain recordings can support meaningful decoding of typed sentence activity. That shifts the frontier. The question becomes less, can any information be extracted, and more, can the whole interface work reliably and responsibly? That shift is the practical implication. The next generation of brain-computer interface debates will not be settled by asking whether a model can produce text from neural data at all. They will be settled by the full stack, sensing hardware, signal quality, task design, calibration, decoding, feedback, reliability, usability, consent, privacy, governance, and deployment conditions. If any one of those layers is missing, the signal may still be scientifically valuable. The decoder may still be impressive. The paper may still mark progress, but the interface is not there yet. That is the difference between a laboratory breakthrough and a practical communication system. And it is the difference between a sensational story that asks us to fear the wrong thing and a story that actually helps us understand neurotechnology. Brain 2 QWERTY does not show that thoughts have become text on demand. It shows that under the right constraints, brain activity can carry enough structure for a system to begin decoding typed sentences. The breakthrough is not that the brain signal is the interface. The breakthrough is that we can now see with much greater precision what an interface would have to become. For more evidence, first biomedical analysis, subscribe to Quantum Convergence.